Today's video lecture is going to cover DNA replication. We need to make copies of our DNA so that we can make new cells within our body. Now, one of the enzymes that helps with DNA replication is called DNA polymerase. You need to understand what a replication fork is, the point where the DNA, two strands of DNA, will be splitting apart. We're going to talk about new and original strands, because we don't just take a photocopy of one strand of DNA and make a second. Okay, we're going to make, because they're called complementary strands and we have base pairing, we're actually going to take the two strands, split them apart, and make a new one attached to that. So we'll see how that happens. We're going to talk about what happens and what direction the replication goes in. And finally, there are a few differences between eukaryotic and prokaryotic DNA replication. So those differences there. So, first thing is that the copying of the code happens during S phase of the cell cycle. So if you're having issues, please refer back to the cell cycle information that we've talked about earlier and what S phase is. So it happens during S phase of the cell cycle, where we go from one strand of DNA in each chromosome to two strands of DNA in each chromosome. So we go from a single strand, so it looks like half an X, into a double strand, so it looks like the full X of DNA. Now, with this, we have our double-stranded DNA. We have all of our letters in here. And what happens is the DNA will start to split, okay, at one point. When the DNA starts to split, we're going to end up with a couple of things happening, okay? You'll notice that they're all joined together here. These bonds, which the bonds between are all hydrogen bonds, are going to get pulled apart. And as those bonds get pulled apart, we have a structure that comes in called DNA polymerase. The DNA polymerase takes and kind of hovers around a section of DNA. And what this does is it helps attach the new bases in there. So it's going to make sure that C attaches to G. If I can get this to, okay. It's going to make sure that this C attaches to a G. It's going to make sure that this C attaches to a G, and it's going to work its way down the strand. Now this is our new strand of DNA. On the other side, the DNA is going to replicate in the opposite direction. So the DNA polymerase is going to attach down here, and it's going to work and make sure that we have an A attached here, we have a C, and an A attached. And so that what we end up with here is a new strand there, okay? So we have a new strand of DNA forming along the inside here, and a new strand forming along the inside here. And eventually these places where the DNA goes from two original strands and where it splits is called a replication fork. And eventually that will continue to split, okay, both in this direction and down below in this direction. So it will continue to split in those directions so that eventually we get two completely identical sets of DNA, okay? And again, it goes in both two opposite directions based on the direction of the phosphate, okay? So one side, it's going to work in the same direction that the phosphate's in, so because of that, it's going to work in opposite directions. So a couple of things, replication fork being the point where the DNA has split so that it can then add a new piece in. RNA polymerase is an enzyme that helps to add the bases and make sure those correct bases are added. We have one original strand and one new strand that's going to be for each strand of DNA. And it's going to work in opposite directions based on the shape of the DNA. Now, DNA replication happens slightly differently in eukaryotes versus prokaryotes. Remember, eukaryotes have a nucleus, prokaryotes do not have a nucleus. So because of that, eukaryotes have a lot of chromosomes that are in strands, and prokaryotes have one single chromosome that is circular in shape. So, eukaryotes along their chromosomes actually have dozens to hundreds of replication forks that will happen so that it can complete faster. So it starts in multiple places along the line, and then they'll work together so that eventually you get the two complete sets. In prokaryotes, we start with one replication fork. It happens right here usually on one side at one point on the circle, and then it will work in both directions so that eventually it comes back around to the opposite point and the two pieces come together, and you end up with two different circles, each with an original strand of DNA and a new strand of DNA. DNA replication is extremely important through the cell cycle. If our DNA does not get replicated correctly, then we can have issues that happen within our DNA, like mutations, which can then cause issues within our body. So, DNA replication is an extremely important piece within the cell cycle, and it must be regulated so that it happens correctly.